Welcome. Today, we're diving into managing deviations during SIP validation and routine operations. I'm Dr. S.K. Singh, and together we'll explore how to handle unexpected changes effectively, ensuring smooth processes and reliable outcomes. Let's get started. Let's start with an introduction to sterilization in place, or SIP. It's a vital process that ensures pharmaceutical equipment stays sterile throughout manufacturing. Even with strong controls, deviations can happen during validation, routine operations, requalification, or maintenance. Because of good manufacturing practices, every deviation needs to be documented, investigated, and assessed for how it might affect the product. Then, it must be resolved through a solid corrective and preventive action system, or CAPA. This keeps everything safe and compliant. When we talk about the regulatory framework, we're looking at a complex network of global authorities. Key players include the European Commission, the US FDA, the World Health Organization, and international bodies like the ICH and PIC-S. These organizations set the rules we follow. At the heart of those rules is a clear expectation. Manufacturers must have a formal system to manage deviations. That means spotting problems, digging into their causes, fixing them, and making sure they don't happen again, all to keep sterility assurance rock solid. Let's talk about what a SIP deviation actually means. It's any time something strays from the approved procedures or expected conditions during sterilization. That could be the temperature dropping below the validated limit, the sterilization hold time not being met, or steam pressure fluctuating unexpectedly. It might also involve sensor malfunctions, failed biological indicators, interruptions in data recording, or alarms going off during the cycle. Importantly, every deviation has to be carefully evaluated to understand its impact on sterility assurance. When we talk about SIP deviations, we're looking at three main types: critical, major, and minor. Critical deviations hit sterility assurance directly. Think of things like failing to reach the right sterilization temperature or a biological indicator not passing. Major deviations might not cause immediate harm but still need investigation, like a steam pressure spike or a faulty temperature probe. Minor deviations have limited impact, often involving paperwork issues or delays in reviewing records. Each type matters, but they carry different levels of urgency. When we talk about common deviations during SIP validation, a few issues tend to come up. Temperature mapping failures happen when cold spots show up during the validation process, spots that don't reach the required heat. Biological indicator failures mean unexpected microbial growth appears after the cycle, signaling a problem. Steam penetration issues occur when parts of the equipment don't heat properly. And instrument calibration errors can throw off sensor readings, skewing the data. For example, if the target temperature is 121 degrees Celsius, but we only observe 118, the validation criteria aren't met. When we talk about common deviations during routine operations, these are the events we see most often. Interruptions in steam supply, condensate buildup, alarms from control systems, valves not working right, sensors drifting out of calibration, cycles stopping because of power failures, and incomplete documentation of the process. For example, if the validated hold time is 30 minutes, but the actual hold time is only 24, that's a red flag. It could mean a potential sterility risk that needs immediate investigation. When we assess deviations in sterility information packages, we use a risk-based approach guided by international standards. High-risk issues call for immediate investigation, no delays. Medium risks need a formal assessment to understand the impact. Low risks are documented and monitored over time to spot trends. We consider factors like the product type, how severe the deviation is, how long it lasted, the equipment involved, how easily it can be detected, and most importantly, the potential impact on patients. This helps us classify risk accurately and respond appropriately. When something goes wrong, it's easy to focus on the obvious problem, the symptom. But to really fix it, we need to dig deeper. That's where root cause analysis comes in. One common tool is the 5 why analysis, where we keep asking why until we uncover the underlying issue. For example, a low temperature might be traced back to a steam valve malfunction, which itself was caused by inadequate maintenance. Another method is the fishbone, or Ishikawa analysis, which helps us explore all possible causes, from equipment and methods to materials, personnel, environment, and measurement. The goal? To find the true root cause, not just the surface symptom. Let's look at an example from a SIP temperature excursion investigation. During a bioreactor's sterilization cycle, the temperature didn't hold steady at 121 degrees Celsius as required. Instead, it dropped to 117 degrees for 8 minutes. The root cause? A faulty steam control valve. This meant sterility couldn't be guaranteed, so the cycle was considered invalid. The immediate response was to repair the valve, verify the fix, and then repeat the entire cycle to ensure safety. Let's look at what happens when a biological indicator shows growth after a steam-in-place validation cycle. This signals a failure, and we need to dig into why. Our investigation focuses on steam penetration, cold spots, where the indicator was placed, steam quality, and the cycle parameters. In one example, the root cause was condensate trapped in a dead leg, stopping proper heat transfer. The fix? Redesigning the equipment and running a full revalidation. It's all about finding the weak spot and making it right. Let's talk about corrective and preventive actions, or CAPA. Corrective actions deal with problems we're facing right now, things like fixing equipment, repeating a process cycle, or revising procedures to stop issues in their tracks. Preventive actions, on the other hand, focus on stopping those problems from coming back. That might mean better maintenance, extra training, or upgrading alarms. Importantly, regulations require us to verify and document that these actions actually work. CAPA isn't just a fix, it's a commitment to continuous improvement. When it comes to documentation and data integrity, we're dealing with the backbone of any quality process. We need specific documents, deviation reports, investigation reports, risk assessments, CAPA plans, effectiveness verifications, and quality approvals. Each one plays a crucial role in tracking and resolving issues. Now, data integrity follows the ALCOA Plus principles. Everything must be attributable, legible, contemporaneous, original, accurate, and complete. Missing or incomplete documentation isn't just a paperwork problem. It can actually invalidate the entire investigation. So, getting this right matters. When managing SIP deviations, there are some best practices we recommend following closely. First, establish clear procedures so everyone knows exactly what to do when something goes off track. Real-time alarm management helps catch issues early, while regular operator training keeps skills sharp. Trend analysis and risk-based investigations dig into the root causes. Verifying corrective actions and conducting periodic system reviews ensure ongoing reliability. Integrating digital monitoring adds another layer of control. Looking ahead, AI will play a bigger role, helping detect deviations faster and predicting failures before they happen. To wrap up, managing deviations and sterilization in place isn't just a task, it's a critical responsibility. We need to catch deviations quickly, dig deep to find their root causes, and assess risks using solid science. Then, we implement corrective actions, keep thorough records, and monitor trends continuously. All of this must meet strict GMP and data integrity standards. Regulators expect us to evaluate any deviations' impact on sterility before equipment goes back into use. When we do this well, we protect product quality and patient safety every step of the way. Thanks for joining me today. If you want to explore this topic further or have any questions, please don't hesitate to reach out. I appreciate your time and attention, and I look forward to continuing the conversation. Thank you. This video was created with SlideSpeak. Visit SlideSpeak to turn your presentations into engaging AI-narrated videos.